Morning everyone. This is GIAS Academy Ramapuram Chennai. We are giving online coaching as well as the offline. The helpline is given here. The coaching is for CSIR GRF UGC NET. This is about the life science. The class is on Unit 2 Cellular Organization. It is part 4. In this class we are going to see the cell wall of, that is the plant cell walls and its composition. What you see here is the plant cell wall. It consists of 3 layers. The first one is middle lamella. The second layer, what you are seeing here, is the primary cell wall. It is flexible. And the third one is the plasma membrane. The middle lamella is made up of the pectin. And the primary cell wall, if you see, there are 3 composition of microfibrils. One is the cellulose microfibril, and the next one is hemicellulose, and the third one is the pectin. They are embedded in all these microfibrils are embedded in pectin matrix. And the most common hemicellulose, what you are seeing here, is made up of xyloglucan, the protein xyloglucan. In grass cell, if you see, this is replaced by the hemicellulose glucano-arabinoxylon. These hemicellulose in grass is replaced by glucano-arabinoxylon. If you see here, this middle cell wall is very flexible and it grows. That growth is called as acid growth. And if you see the composition of this wall, the cellulose is of 35 to 50%, hemicellulose is of 20 to 35%, and lignin is 10 to 25%. So these are the combinations of cellulose, hemicellulose, and lignin. And where this lignin present in the sense, you can see here clearly, this is the cellulose microfibril, and what you are seeing here is the hemicellulose, and these are the lignin molecules which occupies the space which is present between the microfibrils. And what you are seeing here is the microscopic silicate crystals. These crystals are present in grass leaves. These crystals which prevent the herbivorous animals grazing the grass. And if we see another question, what are the question in the sense, what are the enzymes which is present in plant cell and its function? Let us see. The first one is hydrolases, second one esterases and peroxidases. The last one is transglycolases. The function is cut and trim and link wall polymers, cross-link wall polymers. The next question, if you see, what are the structural proteins found in most cell walls and its function? Let us see what are the protein which is present in the sense. Number 1 is hydroxyproline rich glycoproteins, arabinogalactan proteins, glycine rich proteins, and the last one is proline rich protein. And if you take the hydroxyproline protein, these are present in the specialized cell as well as the concentration is higher in the cell corners. If you take this as a cell, the cell corners are richly supplied with this kind of protein, that is hydroxyproline. And another 2 compounds, if you see, that is suberin or cutin. These are 2 polyester-like polymer, and they protect the cell from the herbivores. Already we have seen one, that is microscopic silicate crystals which prevent the cell walls, which prevent the cells from or the leaves of grass from grazing animal. And one more we have seen is the suberin or the cutin which prevent or protect the cells from the herbivores. If you see, there are 3 layers which may found in plant cell wall. The primary cell wall, this is flexible or extensible, okay, and these layers are formed with this when the cell is growing. And the secondary cell wall, if you take, this is very thick layer. This form when the cell matures, this primary wall become very hard and it makes the secondary cell wall. How the cell is formed, we are going to see the cell wall, how the cell wall is forming in the sense, but during the cell division, the cytokinesis takes place. After the nuclear division, the cytokinesis takes place. So this will be the first formation of this is the lamella, that's middle lamella, which forms first. And these microfibrils are held together by means of hydrogen bonds, and they provide a high tensile strength to these microfibrils. What you're seeing here is a small part of the leaf. This is expanded here, and what you're seeing are the cells. Okay, in between the cells you can see a pore-like structure that is called as plasmodesmata. See, it is expanded and shown here. And this is the hole which is present in between then and there in the cell wall. Because the cell wall is very hard, so it won't allow any material to pass through. So why then and there the pores are present. Those pores are called as plasmodesmata. They allow the material to pass out, and they allow the material to come inside the cell, that is in the cytosol. And one more question here it is raised, that is tension wood. What is tension wood? Tension wood is, you can see such type of wood in the pine trees. So they produce a thicker cell wall on the underside of the branches to push their branches upward. This resulting the wood is called as compression wood. And if you see the hardwood trees, they reinforce the walls on the upper side of the branches to pull their branches up. This is known as tension wood. So if the question asks, you can answer for tension wood. And if you see, these are the red algae. Before going to see the red algae, let us see the green algae. These are the green algae. They are coming under the genera. This is coming under the genera Codium, and this is Dasycladus, and this is Acetabularia. These algae possess a special kind of microfibril that is formed from monocell, and they have the polysaccharides in these cells. They have the polysaccharides. Okay, and if you see the brown algae possess a special kind of pigment called as xylanes and alginic acid. And if you see the red algae possess sulfonated polysaccharide. From the name itself, we can get sulfonated polysaccharide in the sense it is present in the red algae. Okay, xylanes and alginic acid in the sense it is present in the brown algae. So keep in mind. The green algae, the genera are the Codium, Dasycladus, Acetabularia. All these are coming under green algae. Okay, you can see the red algae here. Porphyra, Fulzeron are the red algae. Okay, the sulfonated polysaccharides are present in the cell wall of this. And another one compound you can see, that is sporopollenin, as well as the calcium ions are also present in this algal cell wall. What you see here is the diatom. This is also coming under the group of algae. There is a special kind of acid present in this diatom, that is silicic acid, or it is called as orthosilicic acid. And the formula for this chemical formula is H4SiO4. The acid is polymerized intracellularly, and then the wall is extruded to protect the cell. Can you see the protection of the wall? This is extruded to protect the cell of the diatoms. And here what you are seeing is the true fungi. And this fungal cell wall is made up of chitin polymer chain. Chitin polymer chain. This is the structure of, chemical structure of a unit of chitin polymer chain. Here, just see and note this one, the structure of this chitin polymer chain. This may come as a question. Then the cell wall, if you see the true fungi, the cellulose is absent. Instead of the cellulose, the chitin polymer chain is present. And this, if you take the plasma membrane of the true fungi, it consists of three layers: a chitin layer, a layer of beta 1 3 glucan, and a layer of monoprotons. Some fungi are listed under the protist, that is, one is oomycetes. They possess chlorophyll as well as ionophil. With that, they produce their own food. So they are now coming under or classified under the heterocons because they are autotrophic brown algae as well as the diatoms. Okay, and if you see this one, this is protist. This can also produce their own food. And these oomycetes cell wall contain amino acid hydroxyproline, which contain the amino acid hydroxyproline, which is not found in the fungal cell wall. So only they are coming under this heterocons. Dictyostelids. This is another group of fungi. What you are seeing here is that fungi. They have a reproductive stalk and a sporangium. And these reproductive stalk as well as the spores formed at the apex possess a cellulose wall. Okay, this contains the cellulose wall. The spore wall has shown to possess 3 layers, the middle of which is composed primarily of cellulose, and the innermost is sensitive to cellulase and pronase. So keep in mind these enzymes, cellulase as well as pronase, which is present in dictyostelids. Okay, dictyostelids. Thank you. Thanks for watching this class. Share this video to your friends, those who are preparing for the CSIR NET